Do is take a swab sample of a cold sore or an ulcer. So here's our sample right there, and that sample would be applied onto this slide right here. So let's smear this sample onto the slide right over here, and then what you'll see after adding a stain, this could be either a Giemsa stain or a Wright stain, so don't worry about those names, but there's a specific dye that's added to this. So I'll just write up here a dye of some sort is added. And what you should see if this patient has herpes are these giant cells. These are supposed to be epithelial cells from the swab of the lip or from the genital. So that's one giant cell. So I'll write down here we see a couple of giant cells. And there are a few other here. But what's interesting about these cells is that they don't have just one nucleus. There's several of them. So I can count one-ish right there. Maybe here's another one right there. And then there's another one right there. So this cell is multinucleated. So we say that we have multinucleated, and this is suggestive of a diagnosis of herpes. But the problem is this Zank smear is suggestive of a couple other things as well. In addition to being positive for HSV1 and HSV2, this can also be found with cytomegalovirus, or even with varicella, or herpes zoster, which is another type of virus altogether. Which is why this test isn't as commonly done, because usually a history and symptoms can be suggestive of herpes by itself. But even beyond that, another test that used to be done is what's referred to as a cytopathic culture. So cytopathic culture. So I think you've heard the term culture before. I'll describe it a little more in detail below here. But the reason why it's called cytopathic is that what we try to do in this test is to take cells or cytes and kill them. And so that's why it's a cytopathic culture. Pathic you might recognize